Hi, and welcome to 5-Minute Statistics for Clinical Research. My name is Caroline Herborn, and I am part of the biostatistics team at GCP Service International. In clinical trials, the p-value is often the critical value for deciding on the research question addressed. A crucial point in this context that should be determined in the beginning of the trial is the type of hypothesis testing that will be applied. When applying a statistical test, there are always two hypotheses as a basis. The null hypothesis describes the non-favorable scenario where the investigational product would have no impact on the outcome measured. It is this hypothesis that the investigator wants to reject in favor of the alternative hypothesis. The alternative hypothesis is the one describing what wants to be shown. The sticking point now is how the hypotheses are defined. Here is when biostatisticians talk about one-sided and two-sided hypothesis testing. One-sided testing is the case when we formulate our hypothesis in a certain direction. For example, the blood pressure decreases over time under the new treatment. This would be the alternative hypothesis, stating what wants to be shown. The null hypothesis as a counterpart would state then that the blood pressure is not changing over time, or, to cover all cases here, we should add even increases. Depending on to which direction the hypothesis is set up, the probability distribution can look like these. A directed hypothesis is checking the data only in one direction for the fulfillment of the goal, where alpha half is applied on that defined side. The formulation of the hypothesis is crucial here to make sure that the correct test procedure is applied under the assumed direction of the effect. The conclusions to be drawn from the test results in case of a one-sided test are limited though. A two-sided test is a non-directional hypothesis test. It is used when the research question is asking for a general change without specifying in which direction. This implies, of course, that from a significant result it cannot be derived whether the treatment was better or worse, only that it changed significantly. To get back to our example with the blood pressure, in case of a two-sided hypothesis test, the null hypothesis would state that there will be no change in blood pressure after application of the treatment in comparison to the baseline. Whereas the alternative hypothesis would show that there is a difference between the start and the end of the trial. The statistical consequence is that the alpha is split to the two sides of the probability distribution, since within this test the data has to be checked for the two ends. Is it significantly better or significantly worse than the baseline mean? To clarify in which direction the change was given, point and interval estimators can be applied. This two-sided testing therefore yields more information and allows for more precise interpretation of the results. So that is it for today. As you can see, the decision of the type of hypothesis testing can have an impact on the specificity of the answer of your research question. It also has an impact on the error of the first kind, but it can be specified that in general the two-sided test, due to its open question in the end, allows a more specific conclusion about the outcome of the trial. If there are any additional questions, our team of statisticians is happy to help you out. Leave us a message at statistics@gcp-service.com or leave a comment below. If you are significantly satisfied with the content, make sure to subscribe to not miss the next video.